With more than 6,000 small and micro-cap companies listed, if you're looking for the next apple at the earliest stage, then channelchek truly is the orchard. The listed companies support channelchek, so it's free for you, the potential investor, to gain access to institutional quality research from FINRA licensed analysts, advanced market data, industry reports, news, and a growing catalog of videos and webcasts. Make sure to subscribe to our YouTube channel and register for channelchek so you're always up to date on what's going on at the small and microcap data place. Welcome to the virtual Roadshow series presented by channelchek. This Roadshow features Roni Mamluk, CEO of Ayala Pharmaceuticals, NASDAQ ticker symbol AYLA. Following a formal presentation, she is joined by Robert LeBoyer, Senior Research Analyst for Noble Capital Markets, a FINRA-licensed, SEC-registered broker-dealer, for a Q&A session featuring questions that were submitted by the live audience. You can find Noble's research on Ayala Pharmaceuticals on channelchek.com or by clicking the link in the description. With that, I am pleased to present Dr. Roni Mamluk.
Thank you very much, and thank you everyone for joining us today to hear the story of Ayala. So Ayala is a oncology company with two products in clinical development, and both products are products that we are developing as targeted therapies. We call them AL101 and AL102, and both products are targeting the activation of pathways that have been shown to be involved in cancer. One of them is called the Notch pathway, and the other one is BCMA, and I will share with you some more specifics about how we utilize the mechanism of action of these products to guide our development for these candidate products. So our lead product is AL101, is currently in ongoing phase 2 clinical studies in two different cancers. One is called adenoid cystic carcinoma and the other one is called TNBC. And the second product is in another, we are setting up to start a pivotal phase 2-3 study with this product for a cancer called Desmoid tumors. And we are also in partnership with Novartis, combining AL102 with Novartis anti-BCMA targeting agents, specifically with a bispecific antibody, and this is also in an ongoing phase 1 study. So we have our hands full with different avenues and different cancers for which we are developing these two products. All those cancers are rare cancers with very defined and specific patient population that we and the science know that the pathways that the products are inhibiting are activated and are important for the development of the specific cancers. We have an experienced management team, specifically experienced in oncology and in rare diseases, as we are targeting rare cancers. And at the end of 2020, we have reported the cash and cash equivalents of $42.4 million. We have since completed in February a PIPE of $25 million. And this total cash balance is sufficient to support our operations into 2023. So specifically, as we are a company that develops products for cancers, I want to just stop here and go over our pipeline, where every product is, and also mention the upcoming milestones that that are the driver milestones for the company in the coming year. So AL101 is currently, as I said, in two phase 2 studies, one in adenoid cystic carcinoma, the other one in TNBC. In both of these studies, we are screening for patients that have mutations in the Notch gene, activating mutations, meaning those patients that have the best chances to respond are those that are enrolled into the study. And next year, we plan to open another study in a T-cell lymphoblastic leukemia, or TALL, a rare form of cancer in which over 80% of the tumors or the tumors have activation of the Notch pathway. We are intending to report and update data from the first study in adenoid cystic carcinoma. We plan to report in ESMO. We reported last ESMO in September, and then this year again in September out of the ongoing study. Triple negative breast cancer, or TNBC, this study initiated recently, only started to enroll, and we expect that by the end of the year we will be able to report out of this ongoing study. However, potentially only safety data, as the study have only started recently. We are on track to initiate the desmoid tumor with AL102, the desmoid tumor study, mid-year this year. So we are really now working towards starting enrollment. And then, as said, we partnered with Novartis for multiple myeloma. There is an ongoing phase 1 study with multiple myeloma patients, in which recently we have shared, like really earlier this month, we have shared the dosing of, not this month, in April, the dosing have started of the combination of our AL102 with Novartis bispecific antibody, anti-BCMA and CD3. So I've indicated that the cancers we are targeting are cancers that are rare cancers, relatively small patient population, however, very specific with the activation of the pathways. Hopefully, this will translate into indeed efficacy that we are expecting and hoping to see in these in the studies. The numbers that are here are numbers that reflect the eligible patients, and when we are saying eligible patients, I mean those that are in our planned clinical studies. So for adenoid cystic carcinoma and triple negative breast cancers, these are the number for annual incidence around US, Europe, and Japan of patients with Notch activating mutations, and in TALL and desmoid tumors, these are the incidence for the total number of patients, as we do not plan to select for specific patient populations. So although numbers are looking in the thousands, this is a patient population that still would allow us to have a significant market for these products. So I just want to touch at a very high level of how we are inhibiting the pathway and what is the Notch pathway doing in cancer. So Notch is a pathway that in early development is responsible for the differentiation of stem cells and the maintenance of stem cells. And in cancers, many times what happens is that the cells, as part of being cancerous, becoming cancerous through acquiring mutations, are also developing the features of stem cells. So in cancer, when Notch is mutated, there is a dysregulation of the pathway. And indeed, in certain cancers, like in ACC and in TNBC, the pathway was shown to drive the cancer to be more aggressive, and patients were shown to have worse prognosis if they have the activation of the pathway. In order for this pathway to be activated, an enzyme called gamma secretase, here depicted as scissors, need to cleave and cut the receptor. And the small molecules that we have AL101 and AL102 are blocking the enzyme ability to cleave the receptors, and by that actually eliminating the ability of the pathway to work and to activate. And we have seen in preclinical models, and also you will see in some of the data, that that's indeed help reversing and inhibiting the tumors. There are other gamma secretase inhibitors that were developed. The two that are still in development are one at SpringWorks, and the other one that was developed by Lilly and was out licensed to Juno. It's now at Celgene, or that was bought by BMS. And what you see here are two mouse models that show that AL101 is inhibiting stronger the model that is here, it's TALL model, than nirogacestat, which is the molecule that is at SpringWorks, and crenigasestat, which is the molecule that is at Juno. Meaning that at least preclinically, the molecules that we have, AL101 and AL102, appears to be more potent than the other two molecules that are still in development. And now just going through some of the clinical data that we already have and describing the cancers we are working on. So I'll start with adenoid cystic carcinoma. It is a rare malignancy of secretory glands, mainly salivary glands, and it tends to grow around the nerve and spread. Patients that are diagnosed will most times will undergo surgery, and in 50% of patients it will be curative, and the other 50% tumors will recur. And when it's recurring, it's most times metastatic, either locally or goes to metastasis in lung, bone, liver. No drugs have been approved for this cancer by the FDA, and this is mainly because the activity that was shown in clinical studies was very low. Many different treatment modalities were tried, but unfortunately these tumors do not respond to therapy. Even for chemotherapy, there is less than 10% response rate. What you see here are the numbers for annual incidence. Just a minute. Out of this is annual incidence in the US of recurrent and metastatic ACC, and then out of those, 20% will have mutations in the Notch pathway that are activating, and these are the patients we are enrolling into our study. These patients also have a very aggressive form of ACC. One sees here graphs with two lines, one in yellow, which has, these are patients with where the notch is not mutated, and in blue, it's when the notch is mutated. And what you see is the time here is the median recurrent-free survival. So the shorter time, the faster the tumor will recur. And you see that indeed, if you have in the tumor notch mutations, activating mutations, tumor will recur threefold faster than if you have the wild type. And unfortunately, it also translates to overall survival of patients, where you see that patients with notch activating mutations survive a fourfold shorter time than those with wild type. So these are indeed patients that they have a tumor that is progressing fast, and unfortunately, prognosis is poor. We have enrolled patients into our ongoing study, patients with recurrent and metastatic adenoid cystic carcinoma with notch activating mutations and with evidence of disease progression. We have started with a dose of 4 milligrams once weekly. And this cohort is now fully enrolled, and we are enrolling into the 6 milligram cohort. We have gone up in a dose as the safety profile allowed us to do that. And data from this cohort, the 6 milligram cohort, we expect to report for the first time this September. We received an orphan drug designation and a fast track from the FDA for this indication, and that potentially can help us and help us in a more efficient development program for this drug candidate. So here are the responses from the 4 milligram cohort. What one sees here is the waterfall analysis. Yellow bars depicted the patients that had a partial response per the RECIST criteria. And the others between this line of 20% and 30%, these are the patients with stable disease. Overall, we see 68% disease control rate. This is the combination of partial response and stable disease. 15% partial response and 53% of stable disease. And the disease control rate and is also including the stable disease, because patients, as I said originally, are enrolled into the study if they have evidence of disease progression within the 6 months prior to the study. So having stopped the progression of the tumor and having a stable disease is considered to be clinically beneficial. So we are seeing responses. We are seeing these responses as monotherapy with AL101, which was encouraging. And I want to just show you the extent of response in 4 of these subjects that had a partial response. So subject 6, what you see here is a scan of its liver. Where you see my arrow is the liver size. It is a very large liver, enlarged by the tumor, and all the dark areas are tumor. And this is after 2 cycles of treatment, 8 weeks of treatment. You see that the liver is back to its normal size and much less tumor presence. So there is obviously an effect, and a significant effect by the drug or the drug candidate. Here you see the liver of subject 15. The 2 dark areas are tumor or tumor metastases, and you see the significant effect. One metastasis is barely seen and the other one shrunk significantly. Here, this subject has lung metastases, these 2 bulbs here, where you see the very significant effect of the treatment on shrinking those bulbs. And here, subject 35, another subject where the liver is full of tumor, all the dark areas, and you see the significant effect after 8 weeks. So indeed, there is a monotherapy activity, and some of the patients are benefiting from this treatment. Here, these are the list of adverse events or treatment-related adverse events. And you see that the most abundant adverse events is diarrhea and nausea. These are on-target effects of gamma secretase inhibitors, like our drugs. The good news here is that most of these adverse events are grade 1 and 2. Therefore, for oncology, this is what you see here is considered a well-tolerated side effect profile. And this was the basis for why we were able to go higher in the dose to the 6 milligram dose, which is now ongoing. I'm now switching gears to the triple-negative breast cancer, or TNBC. This is an aggressive form of breast cancer. Unfortunately, so far, treatments are effective, but for a short period of time, and patients tend to start treatment and then develop resistance and go through several lines of therapy, but eventually, unfortunately, patients pass away from this tumor. Notch activation was shown by mutations and fusions were known and shown to correlate with poor prognosis. And in about 9 to 16% of the TNBC patients, one can see notch activating mutations and fusions. And these are the patients that we are enrolling into the study. Patients that had gone through 3 or less lines of therapy are those that are eligible to enroll into the study. I want to show you here the basis for why the results and the data that we have generated that guided us to enrich and select for patients with notch activating mutations. So what you see here are graphs from mouse models that are called patient-derived tumor models, or PDX. And these are biopsies of actual tumors that were taken from patients and then implanted on the back of mice, and then treated. The mice are treated with in this case AL101 to see the effect of the tumor. So each graph here represents a tumor from an actual patient. So these 4 different tumors are from 4 patients that had notch activation through mutations or fusions, and these 5 had no activation of notch. And you see the difference in the light blue. This is treated with AL101, and in the dark blue, it's not treated, or treated with a control, with vehicle control. And you see the very significant effect on the tumor models that had notch activation, and much less of an effect in those that have not had the activation. So this is the basis for why we are enrolling those patients that have the best chances to respond. And this is the study design. Patients enroll if the mutations or fusion of notch are detected in their tumor, if they have recurrent and metastatic triple-negative breast cancer, and we've failed 3 or less lines of systemic therapy. And this study have started to enroll a couple of months ago. Here, patients are treated with 6 milligrams once weekly to begin with, and then continue to treatment. And as I said initially, we expect to report data out of this study by end of the year, potentially only safety data, but we'll see how enrollment will go. So I'm now switching to AL102. AL102 is another gamma secretase inhibitor that we have. We in-license both products from BMS. And this product is orally available. And we are developing it for desmoid tumors. Desmoid tumors are, again, rare and aggressive connective tissue tumors. They do not metastasize. However, they grow locally and interfere with organ function, and they cause death, loss of function of the organ. So really, and a tumor that, although it is not metastatic, it's a devastating disease. Patients will go through surgeries, but up to 70% of those patients will relapse after surgery, and sometimes surgeries are impossible because of the location of the tumor. Notch plays a role in the activation that causes the tumor. So in inhibiting notch in this tumor, we do not select for specific patient population, but all patients with desmoid tumors. No FDA-approved therapies for desmoid tumors. The standard of care is limited. So there's actually an unmet need for a good and effective systemic therapy. The gamma secretase inhibitors have been shown to have effect in desmoid tumors. What I show you here are the data from phase 1 studies that were conducted by BMS with both AL101 and AL102, and the phase 1 studies included a handful of patients with desmoid tumors. So for AL102, in the phase 1 trial, there was one patient with a desmoid tumor that achieved a stable disease and some shrinkage of the tumor. In AL101, 3 patients have been treated in a phase 1 study. 2 had confirmed partial response and 1 had stable disease. These studies have been initiated over 5 years ago, and these patients are still today partial responses. So they've still today benefiting from the treatment. An additional patient was enrolled in January of 2017 into an extended access program and had a dramatic response. And you see here the scans. You see here, this is the desmoid tumor, and you see how much it's shrunk. And this patient is also still on treatment. In addition, as I mentioned originally, SpringWorks has an ongoing phase 3 study in desmoid tumors using their gamma secretase inhibitor. They've announced that they will be reporting the data out of this phase 3 study in the second half of this year. They have shown in phase 1 and phase 2 study that there are responses to their gamma secretase inhibitor. So it seems to have promising potential that gamma secretase inhibitors will indeed be a class of potential products that will be an effective treatment in this tumor. AL102 has gone through a phase 1 study at BMS, testing different dosing schedules. So testing daily dosing here, increasing doses, and also intermittent dosing, or what we call weekend dosing, twice a day, consecutive days, and then 5 days off. And what you see here in purple are the 3 dosing regimens that we are intending to evaluate and compare for safety and efficacy in our planned pivotal phase 2-3 study. Here is the study design. It looks complicated, but I'll take you through that. So there is the part A, which is an open label part, where we are comparing 3 dosing regimens to a total of 8 milligram weekly, either daily 1.2 milligrams, so that amount to 8.4 weekly, or 2 or 4 milligram. As I said, the weekend dosing, 2 days off on, 5 days off, to a total of 8 milligram. And these dosing schedules will be compared for safety and also for effect of tumor volume. By the end of this, the part A, a dosing regimen will be selected into part B, which will be a double-blind placebo-controlled study. And FDA, when we discussed this study with the FDA, they've agreed that, pending the results, this may serve as a pivotal study, meaning that the study that based on that, it will be evaluated for approval. And this study is about to begin, and we expect to report data out of the part A, which is an open label, mid next year. So that will be the first time that we will be reporting data. But as I said before, in the second half of this year, SpringWorks will be reporting out of their phase 3 study. So we may be able to see, hopefully, that indeed there is an effect of this class on desmoid tumors. And we are planning and expecting to be second to the market after SpringWorks, with the benefit of their dosing schedule is twice a day with a much higher doses that we are giving here. And we are expecting to be second to the market with a daily dosing or 2 days on, 5 days off, which is a benefit from the perspective of pill burden for patients. Lastly, our partnership with Novartis on multiple myeloma, and there it is the role of gamma secretase in shedding of BCMA. Inhibiting gamma secretase will inhibit the shedding of BCMA, will have more BCMA on the cell surface of multiple myeloma cells, the tumor cells, which is the entity that is being targeted by the treatment that is developed by Novartis and by others as well. So that's the partnership. What one sees here, and I'll focus here, is that in the presence of nanomolar, very low concentrations of AL102, there is a 20-fold increase in membrane-bound BCMA, which is what we are expecting to see. So there is more target for the bispecific antibody. And then comparing with the other two products that are in development as combination with anti-BCMA, in green, it is the springwork molecule that on this cell line and in this mechanism is less effective than AL102 and also the crenigastat. And importantly, what one sees here, it's a study that was conducted by Novartis using cells from 3 multiple myeloma patients. This is ex vivo study. And what you see here is that the lower the number is, the more potent the killing that is induced by the bispecific. And you see that indeed in low nanomolar concentrations of AL102, there is at least 2 orders of magnitude more potent activity of the bispecific antibody of killing of multiple myeloma cells from the 3 patient donors. So actually this is promising because this is what we are hoping, and Novartis is hoping that will be happening with patients in the trial. There is a phase 1 study that is opened, and AL102 is now being tested in combination with the bispecific antibody. Unfortunately, we are unable to provide you expectations for data reporting because this study is run by Novartis. And Novartis, actually speaking of Novartis is we have a partnership with them. They have led the private B round in the company and also joined as investor in the IPO. Lastly, before I finish and discuss it with Robert and answer questions, just to go over what we were able to accomplish. So since we were founded 3.5 years ago, we were able to report already data from an ongoing phase 2 study in accuracy and in ACC and C monotherapy activity in that very tough to treat cancer. We have started the dosing of patients in the triple negative phase 2 study. We reached the agreement on a third study to be a pivotal study for AL102, and we are on track to open this study mid-year this year, and then also started the dosing of AL102 in the phase 1 study by Novartis, so this partnership is ongoing. And then we have a few important milestones this year. In the fall, we plan to report from an additional update from accuracy. And then by end of the year, report out of the tenacity. And these are the key milestones for this year. So with that, I will hand it over to Robert for the questions.
Thank you, Dr. Mamluk. The first question comes from our audience, and someone is asking, what the earliest possible date that an NDA might be filed could potentially be, which based on where you are now, difficult to answer, but just rough time frames, ballpark.
It's very difficult to answer. Where we stand now, I do not see. I cannot answer this question because this is too early. You know, we don't have yet the data from the 6 milligram dose of the accuracy study. So we don't know how this data will translate, whether there is a potential to submit or not based on this data. We don't know. It's too early to tell.
Okay. As an analyst, I guess that's something I'll have to predict. So I'll look forward to making an estimation and answering the question on my own. And in terms of the patent protection, do you expect to get a new chemical entity exclusivity? And
yes.
What would the patent protection look like?
Yes, so actually we do have patent protection on a new chemical entity, a composition of matter, and this is without extension. This is until 2032 and 2034 for the two product candidates. Yeah. So that came from BMS.
Okay. And one of the questions that I have is the notch pathway. Sounds like it's a very good mechanism of action and has a lot of potential in these indications. How common is it in other tumor types or this mutation throughout the body?
Yeah, so what's interesting is that you can see it in other tumors, and we are still evaluating additional tumor types where we suspect that it's not only mutated, but that inhibiting the mutations is a driver. So it can be mutated, but then there are stronger drivers by other pathways, so inhibiting it will not have such an effect like in these tumors. We are doing this work. We have a research team that is testing additional tumor models using these sophisticated patient-derived tumor models, where we are looking whether the fact that there are mutations is also they are acting as driver mutations, meaning treating with AL101 or AL102 will inhibit the tumor, and it is an active effort. And once we find something that we can share with you, then that will be
And just to review the trials to summarize them, my understanding from the presentation is that AL101 is in the phase 2 trial known as Accuracy for adenoid cystic carcinoma, the head and neck indication. Head and neck being a very difficult cancer in general and a difficult field, here's a tumor type with very poor prognosis and very difficult to treat. So this would have a clinical impact on that alone and save lives. And then the Tenacity trial is in triple negative breast cancer, an aggressive form of cancer that lacks the three targets, the estrogen, progesterone, and HER2, that are the common mechanisms of intervention for current drugs. So the triple negative lacks those three, and the Notch pathway provides a new mechanism for therapy for these patients. Correct?
Yes, it's correct, and I would like to add a few words on that. The treatment modalities for triple negative breast cancers are basically chemotherapy based, except if you have the BRCA gene where you have PARP inhibitors as a treatment. However, there are no, there are no treatment. They are very limited, except for PARP inhibitors in a subset of patients. There are very limited opportunities to have treatment that is not chemotherapy based. Therefore, the excitement or of of the physicians, because they said, look, if we have an option like here to tell a patient that she has the opportunity to get a treatment that may have efficacy, but do not carry the side effects and the toxicities of chemotherapy, this is a very good news. So again, we need to see data out of the studies, but at least the important thing here is that this is a targeted therapy, that is not chemotherapy based for this patient population.
Yes, your point about safety and the level of toxicity is the grade 1 and grade 2. The monoclonal antibodies and the kinase inhibitors have severe side effects. So I would see that as a positive. And just the third trial, AL102, about to enter phase 2/3 in the RINGSIDE for desmoid tumors, the connective tissue, the connective tissue metastasis, the connective tissue tumor. Yes, non-metastatic tumor, but still poor prognosis and horrible course of disease and poor survival.
No, I would say it is not. I wouldn't put it as poor survival, but it's what patients experience is that they have these tumors. Let's say they have a tumor here on the muscle. It grows. It's a large tumor. Then it caused pain. You resect the tumor. Sometimes you can there can be an amputation. It's a significant tumor that disrupt the normal function. Many times after surgery, as I said, up to 70% tumors will recur and they need to go to. So it's an ongoing suffering. And in some cases also indeed life-threatening, but not in all cases. Nonetheless, it is a real unmet need for treatment.
Sure. Okay. And you mentioned some of the milestones in AL102 coming up. Could you just review the milestones for the clinical trials over the next 6 months? More chronologically than by
okay.
trial.
Yes, so AL101 will have around September time. We expect to report at ESMO data out of the 6 milligram dose. End of the year, around December, we are expecting to report the data out of the tenacity, the triple negative breast cancer. And then mid-year next year, we expect to report from the first time from the RINGSIDE study, clinical data.
Could you just review the cash balance and your expectations for the cash use over the next year?
Yes, so so we have reported end of the year 2020 to have 42.4 million. Since then, we have added additional 25 million through a pipe in February. So, and this together should fund our operations that we presented here into 2023.
Thank you for joining us for this virtual roadshow presentation brought to you by channelchek. View our YouTube channel for more video content, including C-suite interviews, virtual roadshows, and conference presentation replays. New content is added regularly, so subscribe below to stay up to date. Visit channelchek.com or click the link in the description to access equity research, news, and advanced market data on this and 6,000 other small and microcap companies.